Welcome to our first podcast on the integument, and we will begin with a discussion of the skin. Now, in this first podcast, we're going to be covering the different layers that we have in what we refer to as skin, the outer epidermis, the middle dermis, and the hypodermis. We're also going to distinguish between thin skin, like you might see on your scalp, and thick skin, like the palms of your hand and soles of the feet. Now, as we talk about the different layers, we're going to identify the characteristics that are going to be distinct for each one of the layers, and we'll also talk about all of the different cell types, both covering epithelial cells, secretory cells, such as our sweat glands and sebaceous gland, hair follicles, and our receptor cells that we also have throughout the skin. So the integument is the largest organ in the body. This is one of those trivia questions that often is missed. It counts for 16% of the total weight of the body. And when we mention integument, it's more than just skin. We have our sweat glands, sebaceous gland, hair, and our nails, fingernails, and toenails. Now the skin is more than just simply a covering. And in that sense, it is protective in nature. We are protect from bacterial invasion, from desiccation. We don't dry out. That's what the skin is there for. But our skin and the circulatory system beneath is going to help us regulate body temperature. We have it for reception, which is going to be tactile sensation that we sense from pressure or heat or even some painful sensations. Excretion. Our sweat glands are a way that we can balance our salt levels in our body. And certainly, it's going to be for absorption. Now, this is something that we don't always think about. We think of ultraviolet light being somewhat negative, but ultraviolet light is often used to help us synthesize vitamin D, which is critical in our metabolic processes. But also, ultraviolet light can help break down bilirubin into a form that can be eliminated from the body. So here we see illustrations of thick and thin skin. This is a nice jam-packed illustration showing the different layers that we see present in our skin. Here we can see the epidermis, dermis, and the hypodermis. This is thin skin, like we might see with our scalp. We can see our hair follicles, sweat glands. But this is our thick skin, like the palms of our hands and soles of our feet. So our epidermis actually is going to have 5 layers. The outer, the corneum, lucidum, granulosum are these top 3. Then we'll have the stratum spinosum, and the last we'll have the stratum basale, the bottom layer. So these are the 5 layers that we're going to recognize for thick skin. Some of those are missing over here for thin skin. We have the basale, we have the stratum spinosum, stratum corneum, but we're not going to have the lucidum and the granulosum. Also in this, we can see other cells that are associated with our different layering in thick and thin skin. We'll be getting to those cells toward the end of our skin podcast. So first, just again, sort of taking a big global look at the skin. The outer layer, of course, is the epidermis, epi meaning above. And so this is going to be derived from our ectodermal germ layer during embryonic development. And we already mentioned the 5 layers that we have in our thin skin. And then below that, we're going to have the dermis. The dermis is mesodermally derived. This is that middle embryonic germ layer. And it's primarily going to be our dense, irregular, collagenous connective tissue. We're also going to have receptors that are present down in the dermis. And the dermis and the epidermis are not going to be flat sheets of paper that are abutted against each other, but think of them more like dovetail joint work. You're going to have projections from the dermis interdigitating with projections from the epidermis that lock them together. And this layer where you see this locking together, or this tongue and groove, this is going to be called the reedy apparatus. And the projections from the dermis are called dermal papillae, and the projections downward from the epidermis are epidermal papillae. Now, beneath the dermis, we have the hypodermis, hypo meaning below, and this is not actually part of the skin, but this is our next layer of our body. And it's in the hypodermis we're going to see a lot of adipose tissue, and this is actually part of the superficial fascia that's going to cover the entire body, this connective tissue layer. Now, the last thing to mention are our fingerprints, these very exclusive pattern lines that we see on our fingers. We see them also on our toes. These are referred to scientifically as dermatoglyphs, and we're going to begin to develop these in early fetal development, and they will remain with us and largely unchanged throughout our life. So we're going to start our discussion of skin by first looking at thick skin. Again, in our thick skin, we're going to have all of our 5 layers of our epidermis. We're going to start with the outer stratum corneum and work our way down to that basal layer, the stratum basale. So all of our epidermis is composed of stratified squamous keratinized epithelium. You can see it can be fairly thin, the 0.07 of a millimeter, or it can be over 0.5 millimeter in thick skin. So we have our 5 layers in thick skin. And although your textbook says that thick skin lacks hair follicles, erector pili, sebaceous glands, and sweat glands, you do have some sweat glands in the palms of your hand. And we know that. We can see that in some illustrations. Certainly no hair follicles, no erector pili, sebaceous gland, but sweat glands, you may have some of those in your thick skin. Here we can see why we call it thick skin. When we look at our different layers, here we're going to have our epidermis. This is from this location. This is our basal layer. And this is going to go all the way up to the free border where we recognize our squams, our cells that are remnants of cells that don't have nuclei. So that's our keratinized surface. So this entire layer that we see, this is going to be our stratum corneum. Then we start to get into the lucidum, this darker granulosum layer, the spinosum layer, and this bottommost layer adjacent to our dermis. These are going to be our stratum basale. This projection that you see coming down, that is our epidermal papillae. And you can see this lighter, dense irregular connective tissue projection going upward. That is our dermal papillae, which makes this our reedy apparatus. And just to point out, since we're looking at this picture, as you look in your dermal papillae, occasionally you will see something that almost looks like a fairly large mitochondria. It's going to be somewhat oval in shape. It looks like it might be layered. These are going to be one of our receptors that we're going to talk about as we move on through the rest of our podcast. Thin skin, this is going to cover the remainder of the body other than palms of the hands, soles of the feet. It will contain the hair follicles, erector pili, sebaceous, and abundant sweat glands, but it's going to be missing the stratum lucidum and the stratum granulosum. Now, it's not like you have to recognize those two are missing to realize that we're not looking at thin skin, because this is thick that we just saw. And here we have thin skin, a very different appearance. This is going to be our epidermis, and then we have dermis and hypodermis as we move down. Certainly you can see these large hair follicles, sebaceous glands associated with them, and then we have erector pili muscles here. So it's a very distinctive difference in thick and thin skin when you see them histologically. So now we're going to look at the different layers that we have and look at the histological characteristics of these layers. But understand, these cells of the epidermis, we generally refer to them as keratinocytes. And what's going to happen as keratinocytes mature, we're going to have them formed at the basal layer. This is where mitosis is still occurring. And these cells are going to move upwards until they're shed from the stratum corneum layer. So these cells undergo cytomorphosis, and they are going to change not in just their function, but also their appearance. And they're going to lose the nucleus as we get upwards to the stratum corneum. So we're going to look at those cellular changes that are also happening to our keratinocytes as they move from the basal through these different layers up to the stratum corneum, almost like an escalator as it's moving upwards. So here we can see an illustration of our 5 layers. Stratum basale along the bottom, spinosum, granulosum, and they have stratum corneum. The stratum lucidum is going to be just in this location. Lucidum is not actually a cellular layer, so to speak, but it is a recognizable and identifiable layer that we will look at. So here we see one of our red lines. We've got the illustrations here. Stratum basale, The majority of our epidermis here is going to be spinosum, well, other than what we see in stratum corneum. But all of our cellular living layers, this is going to be the thickest, the spinosum. Here we can see this darker layer that contains a lot of granules. That's our stratum granulosum. Then we're going to have this lighter line where we don't have living cells anymore. The keratinocytes have lost their nucleus. And this lighter or lucid layer, this is going to be the stratum lucidum before we get back into our darker stratum corneum. So the stratum basale, this is where we have our mitotic activity. Now what's interesting is that in the stratum basale, our mitotic activity only happens at night. Now this, my understanding is this is going to protect those dividing cells from ultraviolet damage. So the DNA is more sensitive when it's undergoing condensation, replication. And, well, replication happens, remember, in the S phase. So during this mitotic activity, it's being compacted and separated. So this only happens at night, not during the daylight hours. Now, the stratum basale is also called the stratum germinativum because it is the germinal layer where we're producing a steady supply of new skin cells. And these cells are going to be relatively cuboid in nature. They say columnar, but I've only ever really seen them as cuboid shaped cells right along that basal surface. They're in contact with the basement membrane as well. Now these cells, since they are the basal layer, they are going to have hemodesmosomes that attach to the basement membrane, but laterally they're going to maintain contacts with adjacent keratinocytes via desmosomes, and these desmosomal adhesions are going to stay with them all through all of the different layers as they move upward. Now, other cells that are going to be here in the basal layer that we can recognize that we'll talk about at the end of this podcast are our Merkel cells, which are more like mechanoreceptors, and melanocytes, which are going to be our pigment cells. So here we see a layer through our epidermis. Here we've got our darkly stained stratum basale cells. Here we have one of our other cell types, probably our probably a Merkel cell down here. We've got some other cells that are going to be upwards in this area, possibly Langerhans cells. These are going to be our antigen presenting cells, but we'll get to those also. But along the basal layer, these lighter cells are going to be Merkel cells, and then we'll also be looking at melanocytes that are going to be present here. Now, the stratum spinosum, it is called such because of the desmosomal adhesions between keratinocytes, and now they're going to be three-dimensional in this layer. As the cells during preservation begin to pull apart, the desmosomes are going to hold those cellular processes together, and so the cells look very spiny or prickly. And so we call these cells prickle cells, and if we can go back and just look, you can even see these little lines and just processes extending around all of these cells. This is stratum spinosum in this region. So the cells look very prickly in this stratum spinosum layer. Now remember, when we talk about desmosomes, we have the keratin tonofilaments that are going to be anchoring those plaques from the desmosomes into our cytoskeleton. So those are absolutely going to be present there. And cells in the stratum spinosum adjacent to the stratum basale, they're going to be mitotically active as well. So this lower layer of the spinosum plus the stratum basale sometimes is referred to as the Malpighian layer, which is collectively the replacement layer of cells that are giving rise to new skin cells on a daily basis. So as we move into the stratum spinosum, those light appearing cells that we see that are not our keratinocytes, those are, for all intents and purposes, going to be our Langerhans cells. Those are the antigen presenting cells. So here we have an electron micrograph showing a keratinocyte in our stratum spinosum layer. There you can clearly see these desmosomal adhesions, and you can see the prickly appearance between these different keratinocytes. Here's a nucleus of a keratinocyte, and there's another desmosomes all around these cells. Here's another view really emphasizing graphically all the desmosomes where you see the arrows and the Ds. Here you see a higher magnification look at the desmosoma contacts there. Those are the prickle cells. Now, stratum granulosum, as we're moving further and further upward, we're only going to see this in our thick skin. This will not be present in our thin skin. And this is going to be the most superficial layer that has nuclei. The other layers on top, they will all lack nuclei. So they're going to be dead cells. But even without the nuclei, they are going to be packed with keratin and keratohyalin granules. And these granules and the keratin and the desmosomal adhesions, all that together is going to help to create a waterproof barrier. And this is part of that protective system. We have one waterproof so we don't dehydrate, but it's also being waterproof in that we have a very tight solid barrier to prevent infection. So even in this lower magnification view, we can clearly see this very thick stratum corneum layer. We have the basal layer, spinosum, and then here where you see these darker staining line, that's going to be our stratum granulosum. And even in this one, you can see this faint light line just above the stratum granulosum. That is going to be our stratum lucidum layer. That's coming next. Now, I mentioned before the hair follicles, sebaceous glands, those are not going to be present in thick skin, and they said sweat glands weren't. Here we can definitely see thick skin, and there's a duct of a sweat gland. So sweat glands are definitely in thick skin. Maybe not to the extent that it is in thin, but you see these very often, much too often to say that they're totally absent. Now, the stratum lucidum, this is a fairly thin layer, and it's only going to be present in the thick skin. These cells are filled with keratin filaments, but they're also going to have two other proteins that are present. Ellatin is a refractal and weakly staining type of keratin that's present in these cells. And again, we're going to find these in the palmar and plantar epidermis. That's where we find the thick skin. Palmar is palm of the hands. Plantar is on the soles of the feet. And we're also going to have involucrin, which is present in these keratinocytes, and it's going to be in other types of stratified squamous epithelium. But this is going to come cross-linked with other plasma membrane proteins, and this cross-linking is going to be by a transglutaminase. And this particular enzyme is going to assist in forming this watertight barrier. At the cellular level, it's going to form an insoluble envelope. And so in this sense, stratum lucidum, along with the material being made in the stratum granulosum, is really assisting in forming the watertight barrier for our skin. Here we can see a different type of stain. We have the dermis, a dermal papillae. Here we have an epidermal papillae reaching down. So we've got our basal layer, our spinosal layer. We really have a hard time seeing our granulosal layer in this particular one. This is all going to be stratum corneum, but here we can clearly see this lighter area, and that's going to be our stratum lucidum. Now, the most superficial layer, the stratum corneum, this is going to be present in both types of skin, and it's basically just dead cells. We call them squams, and whenever we have to remove that, that's called desquamation, or the shedding of that skin. Once again, these cells are still retaining their desmosomal adhesions to each other, and that's why they're not shed very liberally. It takes a while for these to come off and a lot of force for these to come off. But they do come off, and that shedding of the skin is another means of protection. As pathogens are trying to gain a foothold on the skin, the floor keeps slipping out from under them, and they are lost as the cells are shed. So here we see another photomicrograph of thick skin. We can easily see our basal, spinosum, even the granulosum, the dark line. The lucidum is hard. You don't know what's artifactual and what's real here, but clearly you can see the stratum corneum and all the multiple layers of our squams that are present and make up that very thick stratum corneum. Now, cells of the epidermis, keratinocytes absolutely make up the bulk of what we see in our epidermis. And they're called keratinocytes because guess what? That's what they make, keratin intermediate filaments, and they're filled with those as well as the keratin hyaline granules. Now, the lifespan of a keratinocyte is 20 to 30 days. Now, that 20 to 30 days, I'm not so sure if it's from the basal layer to when they lose the nucleus, or if it's a month from the basal layer until those cells are actually shed from the surface. But still, a lifespan of a month is pretty quick for most of the cells in the body. So here we can just see the bottom portion of an epidermal papillae as it's reaching down. We have our stratum spinosum keratinocytes. Here we have our basal cells. And then in this layer in the basal, we're also going to have our melanocytes, and our Merkel cells are going to be present here. Langerhans cells are going to be the cells that are located in the stratum spinosum region. And these cells are not rare. 2 to 4% of the cells that you see in the epidermis or in our stratum spinosum are going to be Langerhans cells. And as I've mentioned before, these are going to be essentially similar to our dendritic cells that we've seen in our lymph nodules. These are our antigen-presenting cells. Now, since they are antigen-presenting cells, it's not surprising that they are part of our mononuclear phagocytic system, and so they're related to macrophages. They are also phagocytic. They possess the constant fragment and C3 receptor, so that makes them, for all intents and purposes, the macrophages present in the skin. Now, one way that these can be identified at the electron microscopic level are these inclusions in these granules that contain hydrolytic enzymes, and they're going to look like ping pong paddles or tennis rackets, and these are called Burbick granules. Now, at the light level, you certainly can't see them, but as we look in the stratum spinosum region of our epidermis, and we can clearly see the prickle cells present here. These lighter cells that are present in the spinosum region, these are going to be our Langerhans cells. And you can see the extent to which they exist in the skin. All of these are stained with material that are taken up by phagocytic cells in the skin. And so you can see quite a few of these in any one section that we might see of skin. Now, at the EM level, as we look at the different organelles, you see these handle-like features that are present here. This almost looks like a spoon with a sort of a rounded head on it. But if we look a little bit higher magnification, here again we can see these handles, the head of the racket, and again these are the Burbick granules, these specific crystals that are present inside of our Langerhans cells. That helps identify them at the ultrastructural or the EM level. Now, the Merkel cells, these are the lighter cells. Well, one of the lighter cells. We're also going to see our melanocytes present in this basal layer. But these Merkel cells are going to essentially be mechanoreceptors. And what's going to happen is these Merkel cells essentially are sensory nerve receptors. And so as you impact these with changes in pressure, they are going to secrete neurotransmitters. Now, you're going to have neurons, sensory processes that are adjacent to these Merkel cells, and they can detect this depolarization or this excitatory signal, and that transfers the signal from the Merkel cells to the sensory terminals that are going to send this information then on to the brain. So here we see the illustration. We see these Merkel cells down in our epidermal papillae. You can see the nerve terminals and how they approximate the area of these Merkel cells. At the light level, once again, we can see our spinosal cells. This is an epidermal papillae, and along the basal surface, there we have that lighter cell. That's a Merkel cell. There we have a lighter cell, again the Merkel cell. And at the EM level, when we look at these Merkel cells, they are going to have these processes. They call them spikes that extend up and surround the other keratinocytes. So as pressure is exerted on the skin, it's going to put pressure on these spikes. That causes a change in the membrane potential of the Merkel cell that is relayed down to the nerve terminal. Here you see a bunch of mitochondria all packed together. And here in this nerve terminal, you're going to have basically a synaptic cleft right here. And so the neurotransmitters are secreted from the Merkel cell to the nerve terminal, and that sends the signal on to the brain. Now, our melanocytes are also going to be present in the stratum basale, but these are going to extend processes upward into the stratum spinosum as well, almost like opening up an umbrella. And so I want you to think of them something like an umbrella, not exactly the same, but hopefully you'll get an idea for what we're doing here. So what we're going to have that's going to be characteristic in melanocytes are melanosomes. And the melanosomes are essentially membrane-bound vesicles that have tyrosinases packed inside of them. Now, these tyrosinase enzymes are going to take tyrosine, and these tyrosines are going to be acted upon by that enzyme and converted into melanin. Now, this process of the conversion, guess what? It's activated by ultraviolet light. So the more ultraviolet light, the greater the process of conversion of tyrosine to melanin. Now, these melanosomes, are these factories of melanin, aren't just maintained in the melanocytes, but they can in fact transfer these packages, these melanosomes, into neighboring keratinocytes, and this is going to be by a cytokine type of secretion. And these vesicles are going to essentially cover over the top of the nucleus of the keratinocytes, because remember, that's where you want to that's what you want to protect. You want to protect the DNA in these cells. And so as we look at our illustration, we can see our melanocyte in the basal layer. Here are melanosomes that are being produced. Well, these go into these processes that are extending up in between our keratinocytes, but then portions of these processes get pinched off. And our melanosomes actually get incorporated into the cytoplasm of the keratinocyte, and there you can see where they take up residence, forming an umbrella over the nucleus of these keratinocytes, and thereby protecting them from ultraviolet damage. Now, beneath the epidermis is the underlying dermis, and this is going to consist of two layers, our papillary layer and the reticular layer. Now, the papillary layer, this is going to be the layer where we basically have our Reedy apparatus, our dermal and epidermal papillae. And so this is going to extend from the basement membrane, the layer that essentially forms the boundary between the epidermis and the dermis, all the way into our deeper layers before we get to the hypodermis, where we begin to see our other connective tissue cells such as adipose tissue. Now, we've got all of our types of tissue like we would see in our dense irregular connective tissue. We have our collagens, which are produced by our fibroblasts. We're certainly going to have plasma cells, macrophages, mast cells. These are all present, and certainly type 7 collagen is going to be present. Anchoring in the collagen, our interstitial collagens and reticular fibers, with the basal lamina extending down from the epidermis. So here we see our epidermis. Here we have our basal layer. Our basement membrane will be in this location. Then this is going to be our dermis. We have our papillary layer of the dermis, and then we have our reticular layer of the dermis. You see how these fibers are much finer in nature, and then as we get lower, we get into these deep, coarse reticular fibers that we have present. Now, it's a little bit easier to see here because certainly you can see over this longer range the Reedy apparatus, the dermal and the epidermal papillae. So this is our papillary layer that's going to extend down here to where we start to see these more coarse fibers. So this will be our reticular layer. And then here where we have all of our adipose tissue, our superficial fascia, this is going to be the hypodermis layer. It's quite a distinct change once we leave dermis and get into hypodermis. Now, within the dermal layer, we're going to have some receptors. And the Meissner's corpuscle, what I pointed out to you earlier, that's extending up into that dermal papillae that looks like the elongated mitochondria, that's a Meissner's corpuscle. It's going to be a mechanoreceptor, and it's going to be sensitive to pressure, primarily from touch. Now, in the dermal layer, we also have a corpuscle, a membrane-bound or encapsulated receptor, and this is called the end bulb of Krauss. And this one is sensitive to temperature, and I think that's about all we can really say. Older textbooks said it responded to cold, but maybe it's just changes in temperature again for that. Here we can see our epidermis. We are down in the dermal layer, and where we have these encapsulated swirling type of receptors, these are going to be indicative of those mechano or temperature receptors. This is not in an epidermal papillae, so we don't call this a Meissner's, and it doesn't have a nice elongated appearance to it. So this would be our end bulb of Krauss present in the dermal layer.